Hi, today we are going to talk about culturing epithelial cell, which is cornea. What is epithelial cell? It's a major cavity of the body responsible for the recognizing function of many organs and acts as a model of differentiation and stem cell kinetics, and it's a common cancer rise. Here are the examples of epithelial cells.
Cornea is a clear protective outer layer of the eye. Along with the sclera wide of the eye, it serves as a barrier against dirt, germs, and other particles.
Protocol of corneal epithelial cells. Corneal tissue is placed on collagen and allowed to adhere, and the outgrowth is expanded and propagated in serum-free medium on fibronectin collagen-coated surface. For the materials that used sterile or aseptically prepared, first one, keratinocyte serum-free medium, and the second one, Eagle's minimal essential medium. For the third, Dilbecose phosphate buffered cell line without calcium ion and magnesium ion. For the fourth one, FBS. Fifth one, trypsin and EDTA. Trypsin used 0.05% and for the EDTA 0.5 millimeter. Next, fibronectin and collagen. Fibronectin used 10 microgram per milliliter and for the collagen 35 microgram per milliliter with bovine serum albumin that 100 microgram per milliliter added as a stabilizer. For the last one, BioCoat 6 well plate pre-coated with retinyl collagen type 1.
For the procedure, primary culture step, place corneal epithelial cell up to the circular surface and cut into 20 regular sheets. Turn each corneal segment epithelial down and place 4 segments in each well. Press each segment down gently to ensure good contact between tissue and culture surface. Allow tissue to dry for 20 minutes. Place each drop kgm each segment. Incubate the culture overnight. The next day, add 1 ml kgm to each well. Fibroblast outgrowth minimized by utilizing a serum-free medium and minimize disruption of collagen matrix. Next, remove the tissue segment 5 days after the explantation and add 3 ml of medium adherent cells to continue to proliferate. Confirm monolayer from within 2 weeks. Propagation steps. Feed the culture twice per week at 70% to 80% confidence. Rinse the cells in DPBSA and release with trypsin or EDTA for 4 minutes at 35 degrees Celsius. Stop the reaction with 10% of TBS. Wash the cells, count them and plate at 1 times 10 to the power of 4 cells per cm square onto culture surface coated with FNC. Incubate the culture cell and change the culture medium to fresh medium 1 day after cyclinization. And after complete culturing corneal, extra precautions have to be taken such as shake the contamination, nutrients and many more, and have to be stored in optimal conditions so that the cell won't die and can be passaged for many times.
Now, we are going to discuss on mesenchymal cells. There are 6 main types of mesenchymal tissues, which are connective tissues, adipose tissues, muscles, cartilage, bones, and endothelium. In this part, we are going to talk on adipose tissues. But why do we choose adipose tissues? This is because adipose tissues are widely being used in research that needs deeper understanding on dreadful diseases such as obesity, inflammation, cardiovasculars, and many more. This diagram shows a parts of the body from a rat that we can get adipose tissues from. How to culture adipose tissues? First and foremost, the rat is anesthetized in the box filled with 70% of CO2 and 30% of O2, and it will then decapitate using guillotine. Guillotine is a machine used to cut off the head of the rat. After soaking the bodies into 70% of alcohol, it will then remove the epididymal fat pads. Fat pad here refers as a mass of closely packed fat cells surrounded by fibrous tissue septa, extensively supplied with capillaries and nerve endings. For instance, is intraarticular fat pads. 4 grams of fat pads is added to propylene vial containing of KRBH buffer at 37 degrees Celsius. For your information, KRBH buffer known as crab ringers bicarbonate Hippe's buffer. It functions as a buffering salt solutions, preserving calcium precipitation, and maintaining medium physiology of the pH range. The fat pads are minced into pieces and collagenized solution is added. It will then incubated in shaking water bath at 37 degrees Celsius for 1 hour until the mixture of the cell achieve creamy consistency. Collagenous solutions here refer to an enzyme that breaks the peptide bonds into collagen. It will assist in destroying extracellular structure into the pathogenesis of bacteria. After adding KRBH buffer, the cells is mixed via swirling and gently pass the cell through the nylon mesh filter into conical tube. Cells is washed with KRBH buffer and centrifuge. The infrared form is removed. The process is repeated twice. The cells are finally washed twice with 40 ml of DMEMA at 37 degrees Celsius. The cell is then resuspended from the surface of the medium in DMEMA at a cytocrete of approximately 40%. The DMEM suspension together with the cells are transferred into one of the 6 cm tissue culture dish by using the wide bore pipette tips. Lastly, the cells are placed in humid incubator at 37 degrees Celsius with 5% of CO2 for 1.5 hours.